Today, we will be presenting a research article entitled Non-Targeted and Targeted Metabolomics Profiling of Tea Plants in Response to its Intercropping with Chinese Chestnut. This study focuses on how an intercropping system affects tea quality at the metabolomic level. Before we begin, I would like to introduce our group members.
Talk about introduction. This study is mainly related to metabolomics, which is a branch of system biology. In this study, the researchers uses non-targeted and targeted metabolomics rather than focusing on only one or two chemicals compounds. The study investigated global metabolic changes in the livers. The main focus for this research is to examine how intercropping, which is environmental factor, alters metabolic pathway. Finally, the changes in metabolites are linked to tea quality and taste, including sweetness, umami, bitterness, aroma, and potential health benefits. This helps explain why tea from the intercropping system is perceived to have better sensory quality. Next, objective. The objective of this study is to compare the metabolic profiles of tea livers grown in a Chinese chestnut tea intercropping system with monoculture tea system. Secondly, to identify key metabolites and metabolic pathway using targeted and non-targeted metabolomics approaches. And the last one, to apply computational and statistical analysis tools in order to identify differential metabolites and altered metabolic pathways. For the methodology, this study consists of sample collection, sample preparation, non-targeted metabolomics, targeted metabolomics, and pathway analysis. First, for sample collection, tea livers were collected from two different planting systems. The first was the intercropping system, which is the Chinese chestnut tea intercropping plantation. The second was the monoculture tea plantation, which was used as the control group. Next, for sample preparation, the collected tea livers were divided into two portions. One portion was used for metabolomics analysis, while the other portion was used for chemical and quality-related analysis, such as amino acids and other taste-related components. For non-targeted metabolomics, the analysis was performed using UHPLC-QTOF-MS. This technique was used to detect global changes in metabolites without focusing on specific compounds. The metabolomics data were then processed using principal component analysis or PCA and orthogonal partially squared discriminant analysis, or we call as OPSDA, to identify differential metabolites between intercropping and monoculture tea samples. After that, targeted metabolomics was carried out. This part of the study focused mainly on amino acids, as they play a major role in sweetness and umami taste. Amino acids were precisely quantified using UHPLC-MS/MS, allowing accurate measurement of metabolites that directly influence tea taste. And finally, pathway analysis was conducted. The identified differential metabolites were mapped to KE-GD pathways. The key pathways affected included amino acid metabolism, flavon and flavonol biosynthesis, and phenylalanine metabolism, which are closely related to tea flavor, aroma, and quality. Next, I would like to invite ZHUMAIRA to continue with her part of the presentation.
Thanks, Natasha. Now, let's look at the main result to see if the intercropping actually makes a difference. For the first one is sensory evaluation success. The sensory test confirmed that the tea from the intercrop system, specifically the activity plantation, ranked the highest. It had the strongest aroma and the best taste compared to the monoculture tea. The second one is the key chemicals indicator. Chemically, this tea showed a significantly lower ratio of polyphenols to amino acids. In the tea industry, a lower ratio is the gold standard for a high-quality green tea, meaning less bitterness and more freshness. So, the chemistry confirms the better taste. I will now pass it to ALEEYA to give the conclusion on these findings.
Okay, now for the conclusion on the research, the study demonstrated that intercropping tea plants with Chinese chestnut significantly alters the metabolic profiles of tea plants. Both non-targeted and targeted metabolomics approaches successfully identified changes in key metabolites, including flavonoids, amino acids, and other secondary metabolites. The metabolics differences indicate that intercropping influences plant metabolic pathways related to plant growth, stress response, and tea quality. Overall, these findings provide valuable insight into how agricultural practices affect plant metabolism and quality at the molecular level. Now, I will pass back to ZHUMAIRA.
Thank you, ALEEYA. Now, I will explain the principle and application, which basically the science we use and how it applies to the real world. So, for the first one is metabolomics approach. The core principle here is metabolomics. Unlike DNA analysis, this gives us a real-time snapshot of the plant's chemical products. The second one is two-step strategies. We use a two-step approach. The first one is non-targeted mass spectrometry was used for a broad discovery to find any differences. The second one is targeted mass spectrometry was used for precision to quantify the exact amounts of amino acids. The third one is data processing principle. To handle these complex data, we applied multivariate statistics like PCA and OPLS-BA, which is the principle here is to filter the noise and identify the specific VIP markers that drive the tea quality. The next one is application. The first application is uncovering the delicacy mechanism. This method maps the specific metabolic shifts like flavonoid biosynthesis, which proving why intercrop tea tastes better rather than just guessing. The second one is quality assessment tool. It serves as an objective quality assessment tool why instead of relying on subjective human testing, the industry can grade tea quality based on precise chemical markers. And finally, it applies to agroforestry management. It proves that intercropping is a commercially viable strategy that actively improves product value, encouraging more farmers to adopt an eco-friendly system. I will hand it back to ALEEYA to discuss the advantages and disadvantages of this system.
Based on these findings, we can highlight several advantages of this research. First, non-targeted and targeted metabolomics allowing both broad metabolite discovery and accurate quantification. Second, high sensitivity and reliability in detecting metabolite changes. Third, integration with bioinformatics analysis enables better interpretation of complex metabolic pathways. And lastly, it provides comprehensive metabolic insight. Despite these strengths, there are also several disadvantages that should be considered. First, metabolomics analysis is costly and technically complex, limiting large-scale application. Second, this study may limit the generalizability of the results to other environments. And lastly, changes in metabolite levels were not functionally validated using molecular or genetic approaches. That's all from me. Next, AISYAH will continue to present about databases used in the article.
So now, I will be presenting the bioinformatic workflow used in the article. To understand why intercropping tea with Chinese chestnut trees make the tea taste better, we analyze over 100 metabolites. I will first explain the databases used to identify these chemicals, followed by the bioinformatic tools used to process the data. To identify the 100 differential metabolites discovered in the tea leaves, we rely on 4 major biological databases. The first one is HAMZAH and also MAZLAN. These were used as the primary library for metabolite discovery, allowing us to perform qualitative analysis of the mass spectrometry signals. The second one is PubChem. This will use to verify the molecular structure and chemical properties of the identified compounds. And lastly, the Kyoto Encyclopedia of Genes and the Genomes, also known as KEGG. This was critical for biological interpretation. It allowed us to map metabolites to specific pathways, such as the biosynthesis of amino acids, which explain why intercropping tea increases the umami and sweet flavors in tea. Now, let's look at the tools that make this identification possible. The first one is ProteoWizard. We use this software to convert raw data files from the spectrometer into the XML format, making them compatible with open source analysis. The next one is AISYAH and AMEERA, or R packages. These R-based tools were used to clean the data. XMS perform peak picking and alignment, while AMEERA was used to apply for peak annotation to identify isotopes and adducts. And lastly, Azanian Mass Hunter Workstation. For our targeted analysis of amino acids, we use this professional workstation software to process MRM data with high precision. By using databases to provide biological context and bioinformatic tools for precise data processing, we successfully map the metabolic changes that make intercrop tea superior in quality. Now, we will see the demonstration of KEGG website usage.
And in this video, I will be demonstrating how the KEGG database was used in this research to identify the biological pathways of tea flavor. This study explores how intercropping tea with chestnuts improves its chemical quality. To analyze the data, I am using the organism code CSIN that stands for Camellia sinensis. KEGG's primary function is pathway mapping, which helps us visualize how metabolites work together in a biological system. I am now entering the three key molecules identified in the study, that are L-glutamate, L-thionine, and also GABA. These molecules were found to be significantly higher in the intercrop tea samples, contributing to its superior taste. On the biosynthesis of amino acids map, our metabolites appear as highlighted boxes. These highlights prove that the chestnut trees trigger a flavor factory effect, where the plant shifts its metabolism to produce more amino acids. While tools like XCMS and camera were used to process the raw data and identify these peaks, KEGG provides the biological meaning. It shows us exactly how the environment changes the plant's internal chemistry. By using KEGG, we have successfully mapped the delicacy of tea as described in the article. This concludes our demo.
That's all from us. Thank you.